Gastrulation in frogs is a complex developmental process that transforms a spherical blastula into a multi-layered gastrula, establishing the three primary germ layers: ectoderm, mesoderm, and endoderm. This process involves coordinated cell movements and changes in cell shape and adhesion. 1. Initiation at the blastopore. Gastrulation begins on the dorsal side of the amphibian embryo, opposite the point of sperm entry. Here, a group of specialized cells, called bottle cells, invaginate fold inward, forming the blastopore, a lip-like opening. The blastopore is crucial because it's where the inward movement of cells begins. The dorsal lip of the blastopore is particularly significant, acting as an organizer region that influences the development of the entire embryo. 2. Involution and epiboly. As bottle cells invaginate, they create a space called the archenteron, or primitive gut. Simultaneously, other cells undergo involution, rolling inward over the dorsal lip and into the blastocele, the fluid-filled cavity of the blastula. This inward movement of cells is accompanied by epiboly, where the ectodermal cells spread over the surface of the embryo. The involution and epiboly processes ensure the proper formation and positioning of the germ layers. 3. Formation of germ layers. As cells involute through the blastopore, they migrate and rearrange themselves to establish the three primary germ layers. The cells that remain on the outer surface form the ectoderm, which will give rise to skin and nervous system. The inward-moving cells form the mesoderm, which will form muscles, skeleton, circulatory system, etc., and the innermost layer becomes the endoderm, which forms the lining of the digestive tract and associated organs. The blastocele is reduced in size and eventually disappears as the archenteron expands and the germ layers become more defined. 4. Blastopore closure and archenteron expansion. The blastopore continues to expand, eventually forming a circular opening. As the involution and epiboly continue, the blastopore